Close enough to starting time? I think so. We'll get charging in here. So, let's see. First of all, about the lab. So I checked on your fly bottles this morning. They are looking really good. This experiment is going to work. I just know it. What you need to do next, though, is sometime either today or tomorrow, go in there and clear out the adults. So we've got to get rid of those. I saw some of the bottles were just full of nice little larvae and pupae forming and all that kind of good stuff, so we're going to get some results anytime now. We'll want to see what those F1s look like. That's going to take a little bit of fly time. You're going to have to come in at some time at your convenience to do that. This one, there's no time pressure, so if you start seeing adults, you don't have to rush in and do it. You can give it another couple of days, let a bunch accumulate, then go in for a mega session where you just sit there and count flies. I have posted on the Canvas site a link to a Google Sheet where you can enter the data for your group. So do that whenever. Another thing I mentioned, for this experiment, if you can count 50 flies, that's great. That's good enough. If you want to count more, that's also fine. It's also the case, I noticed some variation in the bottles. Some groups don't have as many flies coming up as other groups. It's also fair game to share out the load. So if you've got a particularly successful bottle, it's got hundreds and hundreds of flies. Yeah, it's the person next to you has 10 flies in their bottle and is looking so sad and disappointed. Give them some of your flies so they can count them. Okay, so lab. Is there anything else I need to tell you about lab this week? You all watched the little video. You know what you're doing, right? Yeah, we get the fun thing of washing bottles. It's just going to be a regular chore. It's not hard, you saw it. And then we're going to get those F2s started. Once you start counting flies, set up those F2 bottles. And then in a couple of weeks we'll have that. And then this experiment will be done. I've also said that we are going to start incrementally working on the lab report. Each group has to turn in a lab report as a group, so it's not individuals turning this one in. And I've asked you to do the first part, which is do a little basic research, find out what these mutants are, write a little introductory paragraph or two, and a little paragraph summarizing what a complementation assay is. And you turn those in to me. I made them due next Monday, so it's not like they overlap with our usual problem sets. And that way I can give you steady, gradual suggestions on improving your lab report. So we want everyone's going to ace the lab report. That's my dream. Okay, let's see what else. Oh, there will be another homework assignment for this weekend. I'll get it out to you by Thursday because I love to ruin your weekends. That's what I live for. Because you ruin my days after when I have to grade them all. But anyway, I have looked over the last assignment, and generally everyone's doing pretty good. I want to comment on one thing, though. I did include a trick question. I think it was question 16. Probably. You all remember this one, right? Yeah, so I had showed you this pedigree, and then I asked you the mode of inheritance. And here's the thing, I gave you a point no matter what mode of inheritance you say. So everyone got the first part right. But then I asked you this question. This is why I gave you points for everything. And I said, look at the pedigree skeptically. Give me two other explanations for the observed distribution that do not involve the stereotypical Mendelian relationship. And the reason I did this is I saw this problem in your textbook and was kind of horrified because what I really wanted you to tell me is, that's pathetic data. Look at that. It's what, 14 individuals, 3 generations. We got 3 instances of some trait. In this case, it's myopia. And if you know anything about myopia at all, it's got a complicated pattern of inheritance. There have been studies, and they've estimated that there are roughly 200 genes that contribute to myopia. So it's one of those multigenic things. This would be really unusual to see such a clear-cut signal out of just such a small sample. So that's one thing to point out about this. What else to point out? Also, another thing I was looking for is, if you noticed, have you noticed that all the college professors here wear glasses? Yeah, we all do. Or some of us wear contacts. This is an example of a pleiotropic effect. It's also an example of an environmental effect. There are so many things that can affect myopia. I blame the fact that I spent my childhood reading comic books with a flashlight under the covers at night, right? Doing that kind of, your parents are right, it'll ruin your eyes. Yeah, and also just the lifetime of just staring at things close up, reading and looking at computer screens and all this, that will induce myopia. So it's also an environmental effect. So I've got a lecture planned for next week, and I'll talk a little bit more about this, about how in many ways genetics has been hijacked by overly simplistic reductionist explanations. A lot of things in genetics are more complicated than can be fit into a single simple Mendelian relationship. Okay, so today, here's what we're going to talk about today. Too much stuff. I've got a whole bunch of things. They're sitting here in this PowerPoint presentation. There's a lot of them. I am kind of confident that I won't get through them all, because I remind you again, do feel free to interrupt me with questions. I am less interested in getting through everything all at once in one big indigestible blob, and making sure that everyone understands it as we go along. So do ask questions. We'll see how far I get here. Then also on Wednesday, I'm planning to have another problem-solving session. We'll just go through and work through problems right up here on the screen, and make sure you all understand how that is done. And yes, it says down there next week, more chapter 4, because I'm so pessimistic about getting through it all today. But also, I'm hoping next week I can start on chapter 5, I think it is, linkage. We've got to start talking about linkage. That's another major factor that affects the frequency of results in a genetic cross. Okay, but today, this is where we left off last time. We're looking at blood. Blood is a wonderful example because it's got all these different traits, all these different heritable traits. There's been a long history of studying it because it's fairly easy to assay for incompatibilities. You just do a blood transfusion, and when one patient dies, you know that there was no, you don't do that. Typically, the way they'll test this is they'll take a small sample of blood from a person, and then they'll hit it with specific antibodies that have been isolated against particular types of carbohydrates and proteins on the surface, and see if it agglutinates. So that's what we're seeing here. Okay, so we got all these factors expressed on the surface of our blood cells, and there's a bunch of them. I'm sure everyone's heard of ABO blood groups, but there's others. There's the Landsteiner group, the MN group. There's RH factor. There's secretor locus, which I won't say much about, but I'll just wrap it up here. Everyone who watches crime shows on TV, you've heard of the secretor locus, right? Yeah, okay, you have. I know how you spend your time. Yes, so there is also a gene that affects the expression of these particular genotypes. So that most people are, is it most? I don't know the exact percentage. I think most people are not secretors. That means that the blood type is the particular carbohydrates and proteins are expressed on the surface of their blood cells, but not in other tissues. And secretors is just another factor. Individuals with the secretor locus will also express those in semen and saliva. So you can guess how this shows up in crime shows, right? Okay, we got the ABO group, and then we got this cool one, the Bombay phenotype. We'll get to that in a moment. But there are at least 33 different blood group systems, and they have varying degrees of clinical importance. That means, what that mainly means is, for instance, the really important one is the ABO group. If you get into an auto accident and you lose a lot of blood, they're going to give you a transfusion. That's the first thing they'll check to make sure you get that their blood is compatible with that particular blood system. The others are also of variable degrees of importance. If you are getting a heart transplant, they might go a little further. They'll check a whole bunch of other phenomena to make sure that they're compatible with the heart you are receiving. But ABO is the main one here. Another factor to explain here, we just talked about ABO. You got type A, you got type B, you got type O. Those are different alleles. There's 3 different alleles for a single locus. And I think I explained this last time, that's a fairly common thing. That the number of alleles is much greater than just 2 in most cases. It's just that any one individual can only carry 2 at a time, assuming they're diploid. So when we describe the relationship here, the word we often use is codominant. Now, last time we also mentioned incomplete dominance, right? The example was the snapdragon, where you've got red flowers and white flowers, and they have progeny and they blend together, and you get pink flowers. You get an in-between state. Codominance is different in the sense that it says here, you don't see an intermediate phenotype, you see both of the alleles simultaneously. So if you have type AB blood, you are expressing both the A antigen and the B antigen. Okay, and this one, MN is another blood group that's floating around out there. It's one of the older ones. It's called the L locus after Landsteiner, who discovered it. Landsteiner was a turn-of-the-century scientist who discovered a whole bunch of blood groups. I think that's what he did all day. He sat there with blood samples and mixed them and said, oh, this one coagulates. Oh, this one stays running. It just works all out that way. Okay, so we got 2 types. We got type M and we got type N. So as you can see here, here's the genotype. And again, remember, you can only have 2 alleles at a time in a normal diploid individual. So we got LM-LM. That's the homozygous condition. That's type M. We got LN-LN. That's also a homozygous condition, which yields type N blood. And then we have the heterozygote, LM-LN. And there the phenotype is MN. Okay, so just a standard codominant condition seen here. Another one is the RH factor, and you might have heard of this one too. So,
are we able to go back to the slide?
Yes, of course we can.
Thank you.
Just transcribing it. Okay, that's good. Yes, it's important to keep good notes, so you never have to listen to my video. Okay, so this is just one of many blood systems. It just exhibits that property of codominance pretty clearly. Next, we're going to go to the Rh factor. Rh is short for rhesus. It was identified in rhesus monkeys. And what the Rh factor does is it's another source of incompatibility. An individual who is Rh negative, if they are hit with Rh positive blood, you transfuse Rh positive blood into them, that individual is sitting there with possibly antibodies that will cause agglutination of the transfusion. And we don't want that. Ready to move on?
Yes.
Okay. So here though is a really common example, relatively common example. There are other ways to get Rh positive blood into your system. So here what you see in this illustration is, here's Dad, he's Rh positive. The Rh positive is a dominant allele. Let me qualify that a little bit, because the Rh factor turns out to contain multiple components that are tightly linked, and we haven't talked about linkage yet, so we'll just say, okay, they just sort of appear to be one single locus, but it's multiple loci. Just so I don't spread misinformation here. Anyway, we've got a guy, he's Rh positive, and the mother of his child is Rh negative. That means that their child could be Rh positive. And normally, the mother will bear this child to term. There is no problems, because there is this massive barrier preventing the flow of blood from infant to mother during the pregnancy. But typically what happens is at birth, if you ever get to go through a childbirth, let me just warn you, it's messy. There are fluids flying all over the place. There are things rupturing. There is blood. Oh man, so much blood. I got to watch three of my kids being born. And I don't know how my wife did it. Anyway, so it's a really messy process. And basically what this means is during this process, it's really easy for fetal blood to get mixed up into mom's circulatory system. And if the fetus is Rh positive and mom is Rh negative, that can mobilize antibodies. So the immune system is going to charge in there to clear this out. Now the thing is, Rh positive in itself doesn't generate a really strong immune response. So the first time you get away with it, baby's born, mom is infected with Rh positive antigens. She makes an immune response and gets rid of them, clears them all out. Everything's great. But then, just as we are experiencing right now with the pandemic, you get exposed to an antigen, like in your vaccines, and your immune system is now primed and ready to make lots and lots of antibodies if the virus comes along. Same thing. What if mom has a second child by this same wicked Rh positive man? Oh man, then you could have another Rh positive child. And then what happens is mom's antibodies attack the poor second child. And they can be born with this thing called erythroblastosis fetalis, or hemolytic disease of the newborn. And that means that when the child is born, they are often anemic, they're weak. You can actually take blood samples from them and see the blood cells being ruptured and destroyed as fast as they're being made, just about. And the main treatment is immediate transfusion of the newborn baby. Oh, that's a traumatic thing to do to a little baby, isn't it? Not only is he shocked with facing the world, but you got to give him this massive transfusion of blood to get his circulatory system up and working. Another treatment, though, One that's often carried out is OBGYNs should know about this. You got an Rh negative mother. You look at the baby, oh, it's Rh positive. And then what you immediately do right after the birth of the first child is give mom a massive dose of something called Rogam, which is the antibody against the Rh factor. So the whole idea is you swamp out the response to the Rh factor really fast with massive doses of antibodies so that mom's immune system doesn't feel any need to make more of it. So, just something to watch out for. Yeah, so pregnant moms should always be tested for the Rh factor, and pregnant dads, responsible dads, also tested for this. It's kind of a routine thing in pregnancies nowadays. Okay, so Rh factor matters here. Here's the ABO blood group. One important concept here is when we say an allele is dominant or recessive or codominant or exhibits incomplete dominance, it's always in relationship to another allele. You can't simply say this allele is dominant. It's dominant with respect to some other allele. So in this case, that's really obvious with the ABO blood groups. So type A is codominant to B. And at the same time, type A is dominant to O. You see how those can coexist? So you can have multiple relationships here. It's all a question of what other allele is present. How is it responding to that other allele? As it says here, the other thing about this is, again, what we're looking at is glycoproteins on the surface of red blood cells. You should remember from way back in cell biology, last semester for most of you, we told you that cells are just bristling with all these complex molecules on their surface. It's just not naked membrane sitting there. They got all kinds of proteins and glycoproteins and sugars and all this stuff just hanging off of them. So every cell is surrounded with this layer of these complex molecules. And isoagglutinin, that's what the I stands for, is isoagglutinin, is just one of many factors that are found on the surfaces of cells. And they work in combination. So here's just the possibilities. And again, just using simple Mendelian reasoning, you should be able to deduce all of these, right? So an individual who is of type A can either be homozygous for A or heterozygous for A and O. In which case the O is recessive, obviously. You don't see it. As it turns out, type O is just the absence of a particular carbohydrate on the surface of the cell. So those are all type A. Type B, same thing. You can be homozygous for B, or you can be a heterozygote with B and type O. If you are type AB, that's actually the rarest condition. If you're type AB, that means you have both of those, and you will express both, and your phenotype will be blood type AB. Type O means you're not expressing any of those. You're not expressing A or B, so you've got to be homozygous, and your blood type is type O. Okay, what this means, from a medical standpoint, you'll sometimes hear this individual referred to as the universal donor, because they don't have that particular epitope on their molecules. So you can give somebody who's type A blood a transfusion of type O, and they're fine. There'll be no isoglutenin reaction. Okay. Type AB is often called the universal recipient, because you can give them type O blood, again, nothing for them to react to. You can give them type A blood. They've already got the type A antigen on all their cells, so they're not going to mount an immune response to that. Or you can give them type B, same thing. They've got the type B antigen, so they're happy with it. You can't do that with type A and type B. So if you're type A, you cannot get a transfusion from type B people or type AB people, right? Because they both got the B antigen, so it'll be a cross reaction. You'll get an agglutination, which is not good for healthiness. All right, so you see the relationships there? Let's look a little bit at how this actually works. Oh, look at that colorful diagram. Yeah, that's so complicated, it just makes you more confused, right? But let's go through it, we'll figure it all out. So, on all of your cells, you have this particular glycoprotein. It's universal, all of your cells have this. It's called the type 2 unit. And as you can see, there's a protein unit here, and then there's these 3 carbohydrates gone in a chain. So you all have that, all of your cells. What happens next, though, is that there is an enzymatic reaction that puts this fucose group, that's another sugar, that little red triangle, onto that terminal carbohydrate there. So we're going to add fucose to that. And that creates something called the H antigen. If you are type O, you stop right there. That's all you do. You got this little structure here with the fucose there and these other 3 carbohydrates on there, and that's it. That's type O blood. In addition, though, if you're type A or type B, you have one of 2 other enzymes called A transferase and B transferase. I guess they got bored with coming up with complicated names. So A transferase will transmit a different carbohydrate to the end up there, and B transferase will stick with this one, our little circle there. So we're just going to make a little elaboration on this surface glycoprotein. If you are type AB, you have both of these transferases. And when they go into action, they don't stick both of those onto both a little yellow square and a little yellow circle onto it. You'll get a random mix. So half of your H antigens will carry my yellow square, half will carry the yellow circle. Okay, makes sense? Yes?
Is it always, like, if you are AB, is it always a 50-50 mix of which antigen it has, or?
Ooh, that's a good question. I don't know. I kind of suspect it would be 50-50. We're dealing with large numbers of molecules, and you're making both enzymes. So yeah, I kind of suspect it would be 50-50, just statistically. Yes?
So if you got, if you were AB and you got blood from someone who's A, would you just come off A? Or?
Oh, so if you're type AB, you have both of these, right?
Mm-hmm.
And somebody who is of type A blood would have just this kind. But that looks just like your bloods. That's half your blood is just like that. So you don't have any antibodies against this. Does that help? Does that make sense?
So then would it not be 50-50 anymore?
Oh, if you're type A, then you're making 100% type A blood.
I think she's asking when you get a transfusion if you're AB and you get a transfusion of either A or B, how does that like work to offset the like 50-50 ratio?
Oh, then it doesn't matter.
Okay.
All that matters here is, do you have an immune response to the particular carbohydrate on the surface? And we're saying if you're type AB, you are conditioned to not have an immune response to either of those.
Okay.
Yeah, so if you get a fresh transfusion of type A blood and you're type AB, you could make the argument, well, yeah, if I sample your blood now, I'd find maybe a large fraction is now type A only, but it wouldn't make any difference to your immune system. Okay. Yeah, so this, if you're looking at this, you're saying to yourself, that is, if you accepted everything I said last time, you're saying to yourself, hey, that looks like epistasis. How many of you said that to yourself? No, not yet. We'll work on it. Yeah, so this is an epistatic pathway. We got a chain of enzymatic reactions to produce an end product, and you could imagine mutations that affect any step in this process. So here's an example of that. This is a curious thing that happened. A woman in Bombay was found to have some unusual blood characteristics. So first of all, she's blood type O. That's not unusual. Really common to be blood type O. But then, look at this second point, just by itself. They checked her parents, and one of her parents was type AB. How do you give birth to a type O child when your blood type is AB? They're either going to get the A allele or the B allele, right? So this is a curious thing. Now, this third point is more bewilderment, because her husband is type A. So if she's type O, you should be able to have children of type A or possibly type O. But she has 2 children of type B. How did that happen? How would you explain that? Adopted. Yeah, maybe she was adopted. That's possible. Yeah, maybe one of the people, maybe the person she thinks is her father isn't. If we want to be even more nefarious in the way our mind is working. So there's lots of possibilities there. That third point, though, is hard to explain. She's type O. And she's got 2 children of type B. Are you going to suspect her of philandering around? Ooh, yeah. Man, you guys have nasty minds. So yes, those are possibilities to think about. They worked through it all, though, and they just said, no, that's not the case. She had her blood type thoroughly scrutinized, and of course she's sitting there insisting, no, I never did any such thing. So how do we explain this? Well, it turns out epistasis is going to explain this. So here's what was going on with that woman. She had a mutation in the H gene. There's that gene here at the beginning that sticks a fucose on this carbohydrate. That enzyme was not working in this woman. So all of her blood cells were covered with the type 2 unit, this one, but didn't have any H antigen. And if you don't have the H antigen, this one, the transferases don't recognize it, and they don't stick their respective carbohydrates on that. And that means that she looks like type O from the outside. Yeah, so she's actually this type, You might speculate, though, that, okay, her husband was type A, she's type O. Her husband could not possibly have passed on the A transferase. Or, well, no, he could have passed that on, but he couldn't have passed on the B transferase, which suggests that if this woman had a functioning, it's called the H gene here, if she had a functioning H gene, she would probably be type B blood. Yes?
So does that mean that, like, genotypically she is type B, but phenotypically she's
Ooh,
type
what an
O?
interesting question. Yes, if we had a blood test that looked for the transferases, we might say, yeah, she's got the type B transferase, so she's sort of, she ought to be type B, but the blood typing is looking at the final phenotype, the end result of this epistatic pathway. So no, type B, blood type B means you have this complicated structure, and she doesn't, so she's not type B. Okay. Again, this is epistasis, just like I mentioned before. And there's the definition. It's just the regulation of one gene's action by another. So if I just step back from it, I'll come right back to this. What we're saying is the action of these transferase genes is dependent on the action of this H gene, providing the H antigen for them to work on. So that's classic epistasis. All right, there's a couple other words here. A mutation in ARB will lead to the failure of C to form. That's what we just saw. If you don't have the transferase and you don't have the H antigen, you won't make C. What we'd say here is A is epistatic to B, and B is hypostatic to A. One other way to think of this is epistatic means it's upstream. So A is upstream of B, and B is downstream of A. That helps you think of it better. We are working on some epistatic interactions in the lab right now. Okay, so we're looking at the brown and scarlet genes, and those are epistatic to a third one, the white gene. Yeah, we haven't talked about white. One of the first mutations found in Drosophila was a gene called white. An allelic form makes the eyes totally white. That gene is not what we're looking at here in our lab. We're looking at these other genes, but they all interact to produce the final color of the eye. So when you start reading the flybase, you start looking at those things, you might notice that they say specifically what the allele scarlet does and what the allele brown does. And they'll also casually mention white in there. These are all part of a complex, a multigenic complex of genes that interact to produce the wild type color of the eye. And I'm gonna review this again. Okay, remember I told you that your assignment in lab this week is to write the introduction. And I told you, get on flybase, figure out what brown and scarlet do. That's part of your introduction. The other part is explain what this epistasis thing is that we're looking at. What is the importance of a complementation assay? So here I'm going to spell it out for you. Oh man, I am so nice on some days. Anyway, so we've been looking at this. This looks familiar, right? There's our wild type. It's got dark red eyes, color of my shirt. We also saw the brown flies, brown-eyed flies, which are kind of a dull brown. You saw the scarlet flies, which have a bright red. We haven't seen any white-eyed flies, but maybe we will in the F2s. That'll be cool. You'll see. Okay, so the question is, here we got these 4 different eye colors, these 4 different phenotypes, and we're asking, are those all alleles of the same eye color gene? So you could imagine that there's one grand eye color pigment gene, and it's just variations in the expression of that gene that produces the different colors. Alternatively, you could have a couple of genes that cooperate to create the eye color. And so maybe brown and scarlet are 2 different genes, or maybe they're just 2 different alleles of the same gene. We want to know. So we want to find out by using the complementation assay. All right, so here's the explanation of that. So we want to know whether one mutant is an allele of another mutant. And this comes up fairly often in genetic studies. Yeah, back when I was working on zebrafish, in the early years of zebrafish research, we were constantly generating new mutants. We were zapping fish with X-rays and with chemical compounds that cause mutagenesis. And we'd get these results, and we'd have to ask ourselves, is this a brand new gene? Or is this just a variant of an old gene? So you do a complementation assay to find out. Okay, so if we do a cross between 2 individuals, each individual is homozygous for the allele we're looking at. So that's what we see here. So we got an A and a B gene. We're saying, okay, we got this mutant over here. It's got a mutation in the A gene. We breed it to get a homozygous copy of that. The other one's B. We get a homozygous copy of that. If we cross them, and it's 2 genes, they will complement each other. That is, the wild type copy from the B in this case, and the wild type copy from the A in this one will be found in the progeny, in the F1 progeny. So all the F1s will look wild type. Okay, that's kind of what we expect in the lab. Again, not to prejudice your observations. Just look and see what you get. But what I expect, we think that scarlet and brown are completely different genes, 2 completely different genes. And so when you get your F1s, they'll probably be wild type. They'll be this color eyes, right? Dark red. Again, if you don't see that, if you see brown eyes or scarlet eyes, do record it. You don't throw away data. You include that. We'll figure it out later. So just be thorough in recording these things. Now, the other possibility is that both mutations are on the same gene. We're looking at 2 alleles of the same gene. And when we cross those, we will get, in this case, a heterozygote who's got A and B. Right? We get in this case, we're saying GA, GB would be the end result. So there's no wild type allele present. It's 2 mutant alleles. They will not complement. You won't get the wild type phenotype. You will get something else. What is that something else? I don't know. Again, it depends on the relationship between the 2 mutations. If, for instance, this was the case for scarlet and brown, and we did this cross, you might discover all the F1s have brown eyes, and you would say, hey, brown must be dominant to scarlet. Conversely, you might see they're all scarlet, and you say, hey, scarlet is dominant to brown. Or you could say, I messed up in the experiment somewhere along the lines. Please, Dr. Myers, help me figure out what went wrong with this experiment. Yeah, that's a possibility too. Okay, so that's what you're doing in the lab right now. We are waiting anxiously for those F1 results, which will either show complementation and tell us that we're looking at two different loci, or they will show a mutant strain telling us that we are looking at a single locus. Again, I'll just remind you, this is in your textbook. You can look it up and you can see that what the case is here is that BW is responsible for synthesizing gesopterin, this bright red molecule. Actually, to be precise, remember, when you look closely on flybase, you'll discover it's a transporter protein. It's actually not making the pigment, it's helping to pump the pigment from the enzyme pathway that is making it into the eye. And similarly, xanthomatin is the scarlet product. It's making a brown pigment. And so we see these different combinations that can result. As I told you, we haven't seen white, but here is a white-eyed fly. One way you could get white is if you've got a fly that is carrying the homozygous mutant allele on both loci. We might see that in the F2s. So we're going to do an F2 cross, a dihybrid cross, and again, think back. If you do an F2 dihybrid cross, you'll get a 9:3 to 3:1 ratio. 9/16 will carry both dominant alleles. So they'll be wild type. 3/16 will carry just the scarlet allele. They'll be scarlet. Another 3/16 will be brown. And 1/16 will be recessive for both loci. We'll see white-eyed flies. So just anticipate that for our F2 cross. We haven't got those results yet, but we'll see. It's actually kind of cool. You'll see a 9:3 to 3:1 ratio in the lab, just like Mendel saw. Okay, I'm going to throw some other terms at you real quick. Another complication, and I noticed in the problem set that a couple of you had obviously read ahead, because when I'm trying to explain why this particular combination appears, some of you said things like it's variable penetrance or expressivity, which is exactly right. There are many traits that do not transmit cleanly like a Mendelian trait. You may be homozygous for a particular genotype, but you don't express it. Either because of the problem of penetrance, that is, you got the trait, but it doesn't show at all, which may be because this is a multigenic trait, and there's some other gene in your system that is compensating, so it's making it invisible. And the other is expressivity, the range of expression of the mutant phenotype. So you see it to varying degrees. So for example, there is a gene called bar eye, which makes the eyes of Drosophila they look like bars, just little vertical bars. They're not nice and oval. And it exhibits variable expressivity. Sometimes you'll get a baby fly that's got a perfectly normal looking eye. Sometimes it'll be a narrow slit. Sometimes it'll be something in between. So that happens with some genes. There's another gene, antennapedia. It's a really cool gene because what it does is it transforms the antenna into a leg. So you get these flies with legs coming off the front of their heads, which is very neat to see. But it also exhibits both of these properties. So you get a collection of baby flies out of this, they grow up, and you'll see some that have perfectly normal antennae, some that have gigantic legs hanging out, some that have just a little stub of a leg, all these different variables in how they appear. Again, these imply the existence of multiple genes that contribute to a property. Okay, this is going to be kind of related to something we'll get to when we talk about linkage. The genetic background makes a difference. So, for example, in fruit flies, there's a whole bunch of different fruit stalks that are used for flies. One of the more common ones is called Oregon R. It's just a bunch of flies that were collected in Oregon, obviously enough, many, many decades ago. And they turned out to be fairly robust in the lab. So we raised them, and that's the stock that we make mutations in. Okay, but along with that comes a whole bunch of other characteristics of that initial root population. And those affect the expression of the genes. In zebrafish, we've got the same thing, kind of the standard zebrafish line is called the Oregon AB line. And why do we have that line? It's because we discovered early on that there were a number of different populations of zebrafish that had differential sensitivity to mutagenesis. So having this particular line, which turned out to be pretty tough and to thrive under many laboratory conditions, that's what we work with. It's kind of fake. I mean, it's handy if you just want a nice simple model that you can work with. But there are many other lineages of fruit flies and zebrafish that are somewhat different. So we care about genetic background. In that genetic background, we can also have other kinds of genes. There is, for instance, these things called suppressor alleles. What does that mean? That means if you've got a line of flies that carry this mutation for this suppressor, and then you do a cross with your background line that doesn't have it, you might get a result you don't expect. Because the mutant is expressed in this new line, but it's suppressed by another gene, epistatically suppressed by another gene. So it mentions here, suppressor of white. You can have flies that are homozygous for the white mutation. And you're looking at them and you're saying, hey, you ought to have white eyes. And they don't, they have red eyes. Why? Because there's this other gene that's taking on the role of the white gene and allowing the wild type to come through. Okay, another factor these are called position effects. And these are kind of subtle, typically. And I can't say much about them yet because we haven't talked about linkage. But I will later. So these are cases where you've got a gene at a particular location and somewhere close to it in proximity is another gene which can modulate the effect of the first gene. So we get these position effects. You can sometimes get these when you just have a recombination event that moves the particular gene around. Suddenly you get an unexpected result. So like I said, when we talk about gene mapping and how genes are lined up on a chromosome, we'll talk more about position effects. Here's another fun one. Conditional alleles. Yeah, we've been treating these different mutant alleles as if, hey, you get them, you're stuck with them, you got it. But there are lots of examples of temperature-sensitive and nutritional alleles. That is, you got the trait, but it's only going to affect the individual under certain specific conditions. So temperature-sensitive mutations are really useful because there's a whole lot of mutations that are lethal. That is, if you inherit this mutation, you're going to die. And you're not going to have any progeny. But one way around it is if you find a conditional mutation, The same gene is messed up, but now it's temperature sensitive. So you take your fly and you raise it a particular temperature, it's fine. It's going to grow to adulthood. It's going to produce babies with the mutation for you. But if you change the temperature either higher or lower, the fly dies. Such power you have. You can just play with a thermostat and kill these flies. Okay. There are also nutritional mutants. You only see these in certain dietary conditions, and there's a number of these that have been identified in humans. Phenylketonuria is a common, well, not common. It's one we know all about. These individuals cannot digest one of the amino acids, phenylalanine. So if you feed them food that's rich in phenylalanine, it just piles up, and it produces toxic byproducts, and it leads to mental retardation and death. Oh, it's a terrible disease. It's a hereditary condition. It's also fairly common in certain Jewish populations, especially on the East Coast. So, if you're in these Jewish populations, one of the things they have you do before you can get married is get a test for this trait. If you are heterozygous for it, and your wife is heterozygous for it, you will get counseled. Maybe you're not the best two people to get married, but true love, it gets in the way. You say, I love this person, I'm going to marry them anyway. It's Valentine's Day. We have to recognize that people have these kinds of choices they can make. But then what you do is you got a couple of things. You can get testing. You can get fetal testing to see if your child is going to have this, so that you can deal with it. And the other thing is, if you do happen to have a child who's got PKU, you put them on special diets. They're never going to go to the ballpark and just eat ordinary hot dogs. Right? You're going to have to make sure that their food is always monitored and they only get certain specific foods that exclude phenylalanine. And then they're fine. Everything's good. They'll grow up normal and healthy. They can just never eat anything with phenylalanine in it. Another similar one is galactosemia. These individuals cannot consume the sugar galactose. So it causes the same problems if they eat that kind of sugar. And another one that's much more common, and that some of you in this room may have, is lactose intolerance. That is a conditional nutritional mutation. So as infants, we all made the enzymes to digest milk sugar, to digest lactose. And it was just fine and hunky-dory, and you could consume all the milk you wanted. When you're young, you get to eat all that ice cream. It's delicious. But then some of us, many of us, as we get older, that enzyme naturally shuts down. It's kind of freaky and weird that you still want to drink milk when you're 20 years old, right? Even if it does mean you don't get to have ice cream anymore. So anyway, this enzyme shuts down. You can't break down lactose. It causes all kinds of intestinal distress if you do consume it. Now, in many Western societies, also some African societies, they don't have this problem because we have got persistence of the lactase enzyme. So I am just fine with eating all the ice cream I want. Does it show up? Anyway, I can drink all the milk I want, eat all the ice cream I want. I don't have this problem because I'm still making lactase, which is just bizarre. That's for babies, not for us adults, but yeah, I can make it. Whereas other people shut it down and no, they can't digest this stuff. It's also a case where the genetics is a little bit complicated, because one of the ways you can induce this is, if you all right now said, I'm never drinking milk again, I'm not going to have any of those milkshakes at Dawn's Cafe, I'm not going to have any ice cream. We're just cutting all that stuff out. What will happen is your body will gradually shut down the production of lactase. And then when you turn 60 and want to go celebrate with a milkshake, nope, that won't work because you don't have any lactase anymore. Yes?
Can you like build up a tolerance to it again? Or no?
Can you build up a tolerance to it? That's a good question. Because the evidence is kind of mixed there. We have a higher frequency of lactase persistence in American culture because it's really hard to get away from milk products, isn't it? And you've all got the enzyme. You had it as infants. So it's like, yeah, if I constantly get boosted with more milk sugars, it'll induce my intestinal system to keep producing it. And so it's not as severe. But even in those cases, some people, no, just can't overcome it. Yeah?
So I've heard of people not being like fully intolerant, but like they may have like a sensitivity. So
Yes.
is that like an example of like expressivity?
Yes.
Okay.
Expressivity that may also be conditioned by environmental conditions, right? So yeah, it's variable. It's not like everyone is going to take a sip of milk and go running for the bathroom. It's some people, yeah, that's how bad it is. Others, yeah, I feel a little rumble in my tummy and I think I'll stop there. Okay, so these are nutritional variations in alleles. And there's lots of examples of those. In flies, we also have examples of mutant flies that have to be on special diets, which again is really useful in some crosses. Because it's really nice that, you got a bunch of flies you're crossing, and if they happen to get homozygous for a particular allele, they drop dead on the diet you feed them, and you get rid of them. Okay. Which leads into this next topic, lethal alleles. So, you get a monohybrid cross. We're going to produce a 3-to-1 ratio of phenotypes when we cross these. And like the yellow and green peas, you should get a ratio of 3-to-1, right? What does it mean if you get a 2-to-1 ratio?
That one is lethal.
Say what?
That like one of them is lethal.
Yeah, so you can imagine maybe the condition where you're homozygous for this particular allele is lethal, and those flies all drop out before you even see them, or those pea plants, or those people, whatever. So here's an example from mice. This is an agouti mouse. And what does that mean? Agouti just means, if you look at the individual hairs, they're dark at the ends and lighter colored at the base. So you get this kind of pretty attractive layered look to their fur. This is a yellow mouse. It's yellow all the way through each of its hair fibers. So it looks like that over there. If you cross a wild type agouti to an agouti, it's like illustrated up there. That's the dominant allele, so you get dominant A crossed by double dominant A, you get agouti mice out of it. No surprise, right? If you cross yellow by yellow, yellow is recessive to agouti, but it's messier because what will happen is, as you can see there, it's A-A-Y, A-A-Y, you cross those, you will get 1/4 that are agouti. You'll get 1/2 that are heterozygous for agouti and yellow. And you'll get 1/4 that are homozygous for yellow. And those all die. You don't see those. They just disappear from the population you're studying. So the end result is that you'll see 2/3 yellow and 1/3 agouti. Okay? If you take yellow mice and you cross them to agouti mice, like you see over there, Half the progeny will be agouti, wild type and healthy. Half will be yellow and also healthy. So you won't see that effect. Got a question?
Yeah, so the lethal offspring, are they even born? Like, do you see them? And
No, you don't even see them.
Okay.
Yeah, mice are brutal that way. If there is a fetal mouse that is defective in some way, it will typically be restored by the mother. You won't even see it. So that's the case here. Okay, what I wanted to talk about next, but I will stop here, is let's start combining these. Let's do cork line calculations, which will fit in nicely with what we're going to do on Wednesday. Is we'll just do more crosses like this and combine them and see what happens. Okay, go celebrate Valentine's Day or something.